talk about mitochondria and chloroplast. So mitochondria and the chloroplast are very important because mitochondria is the organelle which can produce ATP. ATP is an energy resource for living organism, and the chloroplast is also very important for plants since photosynthesis can happen in this organelle, and then through the photosynthesis, the plants can convert the energy from the sun into the glucose. Let's talk about more the details about these two organelle. So first, mitochondria. The size is very similar to bacteria, and then mitochondria is very common in most of the cells since it can provide ATP energy resource. So a typical cells may contain like a few hundred or to a few thousand mitochondria. So like for example, muscle cells, those type of cells, they have very heavy energy demand. So for this type of muscle cells, they have like even more amount of mitochondria than other cells. And then let's talk about like the detailed structure about the mitochondria. So basically, this is the structure of the mitochondria. So this is like an animal cell. In this plant cell, this is where the mitochondria located. Okay, somewhere here. This is a nucleus, and then this is a chloroplast. But chloroplast only exists in a plant cell. It doesn't exist in the animal cells. So this is the structure of mitochondria. Outside is outer membrane, and then this is the inner membrane. There are two layers of membranes, and in between of outer and the inner membranes there's a called intermembrane space, and then within the inner membranes is a mitochondrial matrix. Okay, and the inner membrane is highly folded to form the projection called crista. This is a crista. This crista greatly increase the surface area of inner membrane. That's where the ATP was produced. And then what else? The primary role of mitochondria is to make ATP. But like ATP was generated in the mitochondria, but mitochondria doesn't create energy. Rather than like mitochondria can convert chemical energies from the organic molecule into the forms that can be used by the cell. So like for example, when we eat the food, we consume the glucose inside. But like cells cannot directly use the energy from the glucose. So the cells have to take advantage of mitochondria to convert the glucose into the ATP, and then ATP is the only energy resource which can be used by the cells. So what the mitochondria do was that mitochondria will break down like glucose, such kind of a molecule, into a simple molecule ones, and release energy that will be used to make the ATP. Like for example, there's some kind of function called cellular respiration. We can talk about later. So basically, what's the cellular respiration? Cellular respiration is just in mitochondria. Then the living organism will break the glucose to generate carbon dioxide and water, and additionally ATP. So this is all this kind of cellular respiration will happen in the mitochondria. And like why, like for example, why why the living organisms need the ATP, because they need to use the ATP as a source of energy to carry out different functions such as like muscle contraction, the uptake of the nutrient, the cell division, and other like necessary cellular process, which are very important for survival of the living organism. Mitochondria also beside like convert the organic molecule into the energy resource. It can also perform other function as well. Like for example, they can be used, they can be involved in the synthesis, modification, and breakdown, and several types of cellular molecule. Like for example, synthesis of a certain hormone requires enzymes. Those enzymes were existing in the mitochondria. Like another, and one of the examples of other function about mitochondria is like and in mitochondria it can generate the heat in some kind of specialized fat cell, like for example brown fat cells. So those type of like brown fat cells can work as a heating pad. Then with the heat generated by this type of brown fat cells, they can help the animals to reviving and then protect the sun, to protect the animals from the cold environment. Then we're gonna talk about the chloroplast. Chloroplast is the organelles where the photosynthesis can happen. So what's the photosynthesis? Photosynthesis is basically plants can capture the energy from the light and use those kind of energy from light to synthesize organic molecules such as glucose. This process is called photosynthesis. We're going to talk about more details in a later chapter. So first, let's look at the structure of the chloroplast. So chloroplast also have like two membranes, outer and inner membranes. This is like outer membranes. This is like inner membranes. And then there's some kind of like intermembrane space between these two membranes. And then the third membrane is this one. It's called a thylakoid membrane. So and then with this type of a membrane, a lot of thylakoid lumen was formed. What's this kind of the thylakoid lumen? Basically, it's flattened and fluid-filled tubers that enclose a single convoluted compartment. So then form this type of like thylakoid lumen. So you can see, so basically the thylakoid lumen are several layers of this thylakoid membrane formation. And then on the bottom, next to the inner membranes, that's other structure called granin. So basically, this thylakoid lumen, they just stack on the top of the granin, and then this is granin, and then this next to the inner membranes, that's another compartment called stroma. But it's outside of the thylakoid lumen, so there are several layers. 1, 2, 3, 4, 5 layers in this chloroplast organelles. And then this chloroplast, these kind of organelles, they belong to some general like plant organelle called plastid. Plastid is coming from the unspecialized another type of like organelle called a proplastid. So there are different type of plastid. Chloroplast is just one type of them. So the difference between different plastid is it's just the bilayer synthetic ability and the type of pigment they contain. So, like for example, chloroplast, which carry out photosynthesis, always contain a green pigment. This called chlorophyll. So because they have a large number of green pigment in chloroplast, so this, and then the plant like green plants, they have a huge amount of chloroplast existing in their leaves. So this type of like chloroplast give them a green color. And there are another type of a plastic called a chromoplast. And the only difference between chromoplast and the chloroplast, just like these several letters. So this chromoplast, it can function in synthesis and storing the yellow, orange, red pigment known as the carotenoid. So compared to the chloroplast, which have the green pigment, so this chromoplast, it has a yellow, orange, and red pigment. So this type of a chromoplast, they always existing in like many fruit and flowers, because a lot of like fruit and flowers, the color is like yellow, orange, and red pigment. So in the autumn, this chromoplast give the many leaves the yellow, orange, and the red colors. So that's the third type of a plastic. It's called the leucoplast. Also, this type of organelle, they don't have a pigment molecule inside. It's used to synthesize and store the starch. So this one type of the leucoplast, which doesn't have pigment inside, is called amyloplast. Amyloplasts are common in underground plant structures such as root and tubes. So basically, plastid was differentiate each other by the pigment existing in the organelles. So different color of a pigment will give a different category. Another interesting that's one thing I found out, like when reading this paper, it's one interesting is like mitochondria and chloroplast. They have a different genetic informations, which are different from the genetic information from nucleus. Like for example, when we're talking about the individual organisms genetic informations, we're always talking about those kind of DNA information from nucleus of that specific cells. And then but with like scientists, they found out like mitochondria and the chloroplast, even though they're just different and separate organelles from nucleus, they also contain their own DNAs. And then they name this kind of the DNA informations, which is chromosome. They found out in the mitochondria and the chloroplast are referred as the mitochondrial genome or the chloroplast genomes, respectively. So which is different from the chromosome found in the nucleus of the cells. That's called the nuclear genome. So they even found out the genome and the most of mitochondria and the chloroplast are like single cellular chromosome. And they compared those kind of the mitochondria and the chloroplast genomes with the nucleus genomes. They found out like mitochondria and the chloroplast genomes are very small compared to nuclear genomes. And like for example, they found out like the amount of DNA in human nuclear genomes is about like 200,000 times greater than the amount in the human's mitochondrial genomes. And then in terms of genes, the human genomes has approximately 20,000 different protein coding genes. But the human mitochondrial genomes have only about a dozen of protein coding genes. So in summary, so basically the genes in mitochondria and the chloroplast, they are very small, like with respect to the type and the numbers compared to the nuclear genomes, they're very small. And then scientists started asking like why this separate and the different like genomes from mitochondria coming from. Then they have some kind of hypothesis. And then so okay, let's talk about that later. I'll talk about like how this hypothesis works later, but let's first talk about the division of the mitochondria and the chloroplast, how it look like. So the division of the mitochondria and the chloroplast are very resemble to bacteria. So basically there are several steps. So first steps, it's because mitochondria, they have a genome, they have a chromosome. The first, the chromosome located within the mitochondria, they were duplicated, like they were from one, they become two. And then after the chromosome was duplicated, and then the mitochondria began to divide by the binary fission. So basically the one whole thing will become like two, okay? And then each division, each copies will have one chromosome which derived as before. So in this way, mitochondria and the chloroplast division are needed to maintain a full complement of this organelle when cell growth occurs following a cell division. So basically, when cell divide, not only like nuclear will divide, nuclear genome will divide. All these kind of mitochondria and the chloroplast, they will replicate, they will divide to supply the enough amount of the mitochondria and the chloroplast during the cell growth. And besides, environment may influence the size and numbers of these organelles. Like for example, when plants are exposed to more sunlight, the number of chloroplasts in leaf cells increases. So okay, then we talk about the hypothesis, like how those genomes in the mitochondria and the chloroplast derived. So basically, there are some kind of like some theory called the endosymbioses, this kind of hypothesis. So this concept is just a relationship in which like small species live inside of large species. So basically, how does this hypothesis was talking about? So he's talking about this is how this chart gives the details about how this process going. So basically, they assumed mitochondria is original from some kind of bacteria. It's called a proteobacteria. It's like very ancient bacteria. So this type of like ancient bacteria, they will go inside of the some kind of the eukaryotic cell, but this type of eukaryotic cells is just like Asian. It's like cells which existing in long time ago. It's called primordial eukaryotic cells. And then after this bacteria emerged inside of these eukaryotic cells, and then a lot of evolution happened, and then a lot of like the other organelle were generated during this kind of evolution. So let's assume like the genome existing in this kind of Asian bacteria will be the genome which be existing in the mitochondria. That's the hypothesis they're going. They are thinking like the genomes from the mitochondria is from the endocytosis of the bacteria outside. The same thing for like genomes in chloroplast. So they are thinking like ancient, tons of years ago. So the primordial eukaryotic cells will endocytose some kind of like cyanobacteria, and then with the evolutions, those type of like early stage of eukaryotic cells can develop and further divided into the other organelle. So the genomes which exist in this kind of type of the chloroplast were coming from the genomes from cyanobacteria. So that's the series. So basically, during the evolution of eukaryotic species, many genes that were originally found in the genome of the proteobacteria and the cyanobacteria has been transferred from the organelle to the nucleus. And then this happened many times through evolution. So that's why like nowadays the mitochondria and the chloroplast has lost most of the genes that still exist in the present days proteobacteria and the cyanobacteria, because nowadays those bacteria and already doing the evolutions, their genome changed with the time, and then but the genomes which existing in the mitochondria will be coming from like ancient times of the early version of the bacteria. So that's why the genome of the mitochondria is different from the nowadays this type of bacteria. Okay. Then we're gonna talk about protein sorting to the organelle. So eukaryotic cells, they have a lot of like their organelles. So proteins is very common organic molecule which exists in almost every organelles. In this chapter, we're going to talk about how proteins will be assigned into the different part of organelles. So first, let's talk about like how protein was synthesized. So basically, the synthesis of the most of the protein begin on the ribosome in a cytosol using mRNA that contain as a template that contain the information for polypeptide synthesis. So this is like this yellow thing, this yellow big thing. It's a ribosome. Ribosome is basically is a complex of the protein and rRNA, right? So this mRNA is a template for synthesize the protein. So basically, the mRNA will go inside, will have some kind of interaction with the ribosome, and then those ribosome will extract the information from the mRNA and make the corresponding amino acid, and then make the corresponding the proteins. So so let's say the cytosol which will provide amino acid, which is the basic unit and the building blocks to making proteins. But this type of protein which made in the ribosome in the cytosomes, they don't have any sorting signals, so remain in a cytosol. So what is the sorting signals? Sorting signals is just some type of like short fragment of the polypeptide. Like for example, in the protein they are trying to synthesize it, that's like about like 6 to 12 amino acid those kind of fragments, they are specialized. Those kind of small fragment, they decide where this protein will be moved to. That's called the sorting signal peptide. So how does this kind of kind of protein sorting was happen? So like, for example, the protein which was designated for ER, Golgi, lysosome, and the other different organelles, they will begin in cytosol, and then they will stop temporarily until ribosome has become bound to the ER membranes. So after this occurs, translation resumed, and the polypeptides and the protein was synthesized into the ER. Like in this way, protein that are like designated for different organelles will be directed into its designer of organelles. This is called co-translational sortings. This chart clearly described how this process happened. So like, for example, this type of protein, they don't have any sorting signals. So they made in the ribosome, in the cytosols, and then they stay in the cytosols. But like, for example, if they have some kind of co-translational sorting to the ERs, so which means this protein they made has to go to ER finally. How does this thing work? How does this thing work? So translation were temporarily stopped, and protein. So during the translation was temporarily stopped, and then this whole complex, including ribosome and the mRNA and the partially synthesized protein, will move to the ER. And then some of this protein contain ER retention signals and remain in the ER. So this thing will attach to the ER, and then after it's reached to the ER, it will keep synthesizing the protein until it's complete. And then after it finish synthesize the whole protein in the ERs, the protein will be like transported in some kind of vehicle, and then this vehicle which contain the synthesized protein will move the protein into other organelle like Golgi. Like here, some of the proteins, they contain Golgi retention signals and remain in the Golgi. The others are sent through the vehicle to the lysosome, like this way. So basically, that's how it happens. Okay. This is another type called post-translational sorting. What is post-translational sorting? So the uptake of most protein into nucleus, mitochondria, and the chloroplast and the other organisms occurs after protein is completely made in the cytosol. So the difference between this type and this type is different, because this type is basically the protein was synthesized in the ER, like that's the organelle. But for this type of this post-translational sorting, so basically the protein will be completely synthesized there within the cytosol with the help of the ribosome, and after it's completely synthesized in the cytosol, they were sent to the nucleus, mitochondria, like different destiny and designed organelles. What's next? Okay, let's talk about more details about how this kind of thing happened. So how does this co-translational sorting happened in the ER, which means endoplasmic reticular membranes. So first, this is mRNA, this is a ribosome. So first, it will generate like a small fragment of the small fragment of the protein, and this small fragment of protein, it has a sequence of about 6 to 12 amino acids that's predominantly hydrophobic and usually located at one end, which is an end, because this type of like this is called the ER signal sequence. This type of ER signal sequence is very important for directing this protein to the ER for further synthesis. Why it's important? Because this ER signal sequence, it's called SPR. It will bind to some kind of the SPR, and then because of this kind of bonding, and it will stop the synthesis of their proteins. And then because this kind of bonding will help to like guide the holding ribosome and partially synthesize the protein into the ER membrane. So basically, this SPR and the bonding is more like give you a GPS, give you a guidance and direction to help you to reach the designer place. So, and then after it reach to the designer ER membranes, this binding SPR will be released, and then the protein translation will continue and continue to make a large proteins. And then this growing polypeptide is thread through a channel like this, and then SPR will go away. And then and another thing which happened is the old, like this ER signal sequence will be cleaved by some kind of enzyme called signal peptidase, and then this modified polypeptide chain will completely synthesize and then released it into the ER. That's how it progressing during the co-translational sortings. So basically, it needs some kind of the sequence to bind to the SPR, and then this kind of binding to help the protein find the direction, which can direct into the right place, and then keep growing, and then get rid of this kind of signal sequence, and then release the completely synthesized polypeptide, and then this polypeptide protein, which will go to the design place, which is ER lumen. Okay, so alternatively, I should mention about other proteins that are designated for Golgi, lysosome, and other like plasma membrane or other organelles leave the ER are transported to their correct location. This transport process occurs through the vehicle that are formed from one compartment, then move through the cytosol and fuse with another compartment. So basically, when this is like this chart, it's only for like ER protein synthesis. Like for example, for other organelle design protein synthesis, after the protein was synthesized in the ER, and then those protein they will leave ER, and then this protein will be transported during the vehicle, and with the vehicle, it's more like a truck. It can move the protein from the ER into its other designed organelles. Okay, now then we're going to talk about like post-translational to nucleus and peroxisome and mitochondria and other organelles. So most of this protein are synthesized in the cytosol and then take up in their respective organelle. For example, most of the protein involved in ATP synthesis are made in the cytosol, and take up into the mitochondria after they have been completely synthesized. So in order for this kind of process happen, a protein must have a proper sorting signals as part of its amino acid sequence. So how does this? Let's talk about one examples. How a protein is direct to the mitochondrial matrix. So first, like what happened to the ER direct like what happened to the first examples. This protein always have a short amino acid sequence at the N terminal called the matrix targeting sequence. So this is the example. So first, so during the ribosome synthesis of the protein in a cytosol, so a short fragment of polypeptide was synthesized, and then this called chaperone. Chaperone is some type of like another protein. This kind of protein, like this protein, can keep the big ring unfolded, and then beside for this short fragment, this have a matrix targeting sequence, right? And then this matrix targeting sequence, they are part of like small fragment of amino acid. It's very important. It's gonna bind to the some kind of receptor proteins, and then after it's binding to this kind of receptor proteins and the shutdown, those proteins will be released. The protein was transferred to a channel into an outer membrane. So it's binding to a receptor, and then this is like protein channels in through the membranes. So it will go through here. And then first it's into the outer membrane. When it's going inside, and then its protein is go further, go to the channel into the inner membrane, and into the inner membrane. So then protein will transfer from like this is cytosol. This is like a cytosol outside of the cell, and then goes through those kind of membranes. Now it's inside because all the organelles are within the cell. It goes through the channels, move inside of the cell, and then when it's inside of the cells, the chaperone will again bind to the protein. Now these proteins will reattach to this kind of a protein fragments, and then this kind of orange used to targeting into a receptor protein, it will cleaved by the enzymes, and then the protein is completely spread into the matrix. So then in this way, protein will move to the mitochondria. After those protein, it's completely move into the mitochondria, and then the chaperone will be released, and then protein will fold it into the correct three-dimensional structures. So it's very similar to the to these examples. So basically, but this is a little bit like more complicated, because most of the times when this type of the post-translational proteins, they are bigger and more complicated. That's why it involves a more complicated like chaperone to help to unfold the proteins, and then direct it into the mitochondria. So okay, then let's talk about the cells. Okay, let's talk about the cells, like summary of cells from very beginning from the simple ones to complicated ones. Bacteria. First, we can talk about bacteria, because bacterial cells are relatively simple system compared to eukaryotic cells. Bacteria are less small. They are they don't have a like lack of extensive internal compartmentalization characteristic of eukaryotic cells. So it's have like compared to the eukaryotic cells, it's have less amount of their organelles. And then outside, bacterial cells have cell walls, and many species they have a flagella. Those kind of flagella is help bacteria to move, right? And compared with the more complicated cells, animal cells, bacteria have cell wall, but animal cells they don't have the cell wall. And then bacteria, but plant cell, even though they are like more complicated than the bacteria, but plant cell they have cell wall too. And but the chemical composition of their cell wall is different between the plant cell and the bacteria. And the plant cells they don't have like organelle called flagella because they don't have to move. So let's compare. That make a compare the three different type of cells: bacteria, animal cells, and the plant cells. So only the plant cell, only plant cell and bacteria have cell walls, and for fragile and cilia, only bacteria and animal cells they have those kind of fragile and cell and the cilia. And then all these three type of cells, they all have plasma membranes. And then let's talk about the inner structures. Let's talk about the cytoplasm first. For bacteria, the cytoplasma usually they are more simple. For bacteria, they usually have a single compartment inside of plasma membranes. But for animals, they have a lot of like several membrane-bound organelles, right? All these organelles they are surrounded by the cytosol. And then for the plant cells, within the cytoplasma, they are very similar to the animal cells. They have membrane-bound organelles, right? Ribosome existing in all these three cells because ribosome is very important, essential for making the proteins. Proteins are very important for living organisms. Like all cells, they need the proteins to survive, right? And then we compared the difference of the chromosome between three different cells. For bacteria, chromosome only existing in one cellular chromosome per nucleus. So it only like bacteria, it only have one chromosome, and which is circular. And but for animal cells, they have a multiple linear. The shape of the chromosome is linear, and they have more than one chromosome in the nucleus. And the nucleus have a double membranes. And also, we just mentioned about the mitochondria also have the chromosome within the animal cells. Right, the same thing for the plant cell. Plant cell have several and linear chromosomes, and plant cell have both mitochondria and the chloroplast. So both mitochondria and the chloroplast, they have a separate chromosome from the nucleus of the chromosome. And then bacteria, that's very simple. They don't have endomembrane systems, right? They're very simple. They only have like within the cytosol, they only have their one single cellular chromosome. They don't have any other organelle endomembrane systems. But endomembrane systems, they present in both animal cell and the plant cells. And mitochondria will be existing in both animal cells and the plant cells, but bacteria don't have a mitochondria and the chloroplast. Only plant cell have a chloroplast, and animals doesn't have a chloroplast because animals they don't have to do photosynthesis. Okay. Let's talk about the eukaryotic cells in a little bit details. Eukaryotic cells, they are more complicated. They like good example, it's a plant cell and animal cells. They're very common cells in nature. So we can talk about the eukaryotic cells into four part. Those four part, they are interacting with each other. Four part are divided into like nucleus, right? In this nucleus, they have like DNA information, and then cytosol, and then endomembrane system, which composed of the different organelles, and then the other one called the semi-auto or semi-autonomous organelle. Like one good example of this called the mitochondria and the chloroplast. So these four regions, they have different structure and organizations. So that determine that they have different functions. So the first, let's talk about nucleus. Nucleus is the location of the most genome. It's where the place the gene were expressed and regulation. Nucleus is the organization and protection of the chromosome because the chromosome existing in the nucleus. So it's also the site for ribosome subunit assembly. So basically, ribosome will be formed within the nucleus, right? And after the ribosome will be formed in the nucleus, and with the time, it can move outside of the cells, and then this ribosome, it's very essential for the protein synthesis. Then with the transportation of the ribosome from the nucleus to the outside of the cells, then the protein can be synthesized out of the cells. And then endomembrane system is very complicated, right? It's composed of the different organelles. So we're divided into the several different organelles. So endomembrane system, it's composed of the nuclear envelope. So this is a nuclear envelope. It's double membrane, and it's enclosed the nucleus, right, outside. And it's like this is ER, right? It's very common, we have mentioned about the ER a lot. It's an organelle where the protein was secrete and sorting a lot. And then it can help the protein to do other modification like glycosylation. Lipid is a place where the lipid was synthesized. And it's also the organelle where a lot of metabolism will happen. And then it's ER also is a place like calcium will accumulate here. And then Golgi, this is Golgi apparatus. It's also a place protein will be secreted and sorting. It's also the place where the glycosylation is happening. Glycosylation is more like modification of the protein, right? And then this is the lysosome, right? It's a wet place. The organic molecule will be degraded. It's a place for storage of organic molecule. It's also a place. Okay, that's another one plant which have called vacuoles. It's a place like a plant where can accumulate a lot of water. And then there's some like other like small things. I think this is purple thing. It's called peroxisome. Peroxisome is the place where break down the toxic molecule, right? Such as this hydrogen peroxide. It also break down and synthesize other small organic molecule, and then plus membrane, right? Uptake and execution of ions and molecules. So basically, a lot of like the molecule exchange through the membrane. So through the membrane, small molecule can go inside and then come out, right? A lot of like receptor protein receptor will happen here. So it's a place where cell singling and the cell adhesion will happen, right? Then this is like the third part, so cytosol. This is cytosol. It's a coordination of the response to the environment. So, like for example, if some molecule will go through the cell membrane, go inside of the cells. So, because the cells they have a new molecule, so the cells have to be respond to the outside new molecule. So this is where the corresponding happened in the cytosol. It's a coordination of a metabolism. Like for example, if some kind of the chemistry reaction will happen corresponding to the metabolism, is where this cytosol will happen. And then cytosol also is other like some type of protein where the some type of protein synthesis. Like for example, this type of protein is like a protein. Why this protein is very important? Because protein is very important enzymes for degrade the proteins. So basically the cytosol is the location where the breakdown and degradation of the protein will happen. And then in the cytosol, always there's some kind of cytoskeleton and the motor protein that exists, right? So that's where the organization and the movement of this type of other organelle like cytoskeleton happened. And then last part, we're gonna talk about semi-autonomous organelle, which include the mitochondria and the chloroplast we just mentioned about, right? This is the mitochondria, right? Because why they are important? Because mitochondria they can produce ATP energy resource, right? They can help synthesize and modification organic molecule. They can produce the heat. Chloroplast is also very important. They are the place where the photosynthesis will happen. Why they separate these two together with the rest? Because the mitochondria and the chloroplast, they're very important. Besides providing the energy and generate the energy resource, they have their own individual genome to make them separate from the rest, right? For the other organelles, only nucleus have individual genome. The other organelle, they don't have their separate genomes. Only these two, mitochondria and the chloroplast. So we are done with this chapter. Okay. So did I told you to do the homework for this chapter? Have you had a chance to do this kind of homework, or you want me to talk about now, or you want me to keep going?
I don't think I had a chance to do the homework yet.
But let's talk about right now. How about that? For you to review.
Okay.
Okay. Let's do one by one, okay? Since you didn't do it. So the cell theory state that all living things are composed of a cell. Cell are smallest the new cells coming from the pre-existing. I think it's D, okay? The cell theory. All living things are composed of cell. All cells are the smallest unit of living organism. The new cells coming from the cells by cell division, okay? So that's D. When using the microscope, resolution is, it should be D, the ability to observe true. Okay, so the resolution, like when we are trying to, like for example, we mentioned about different type of microscope, right? Light microscope and the electronic microscope, right? So let's really put it for you. So light microscope. Can you see oh, not this way. Not the YouTube. Let's close microscope. Okay. See. Can you see my website?
Mm-hmm.
So, like for the same, like this is different between light microscope and electronic microscope, right? For light microscope, you just see like this is cell, right? This is cell, like this is one particle, this is another particle, right? But you cannot see the details. Like for example, you cannot tell what's different between this small dot and this small dot, right? But with electronic microscope, you can see, even though they're next to each other, like these two, even though they're next to each other, but you can see they're different, right? By structure, right? That's the question is asked about the resolution thing. So electronic microscope, they have a better resolution. What this resolution mean is its ability to observe the two adjacent object as being differentiated from each other, right? Resolution, which means they can distinguish between each other. So that's why D is important, right? The ratio is not about the ratio. It's not about degree, not about the structure, not about degree. It's about distinguishing. So D is important. And then let's talk about the third one. If motor protein will hold in place and the cytosol are free to move, what type of motion would occur when it's B, right? Remember that the things we talked about before? It's like this is let's go to I talk about this with you. Wow. I talk about this kind of chart with you. Maybe you don't remember. Oh, here. Okay, here. Listen. Remember this structure? He's talking about this one. This is a motor protein, right? It's fixed. And then this, and okay, this is motor protein, which is fixed. And then this is called filament. And then it's move, right? This is move. That's what this problem is. It's asked because motor protein, it's held in one place, which means it's stable. And then filament, it's move. That's why we're choosing B, because filament was move. That's how they interact with scalar. That's how motion happened. Age of a foreign part after endo system except. We should choose E, right? Because mitochondria, they belong to the semi-alternate monomers, right? Right? Because they generate ATP and the protein. Essential is important for storage. The structure support should be C. It should be E, A, and C, right? Photosynthesis. It's happened not in a vacuum. It's happened in the chloroplast. Describe two specific ways that protein action are involved in the cell function. Okay, so it's the interaction between the motor protein and filament cause the filament to bend, tubular protein band to each other. It's like the example I mentioned to you before. Okay, these two. Right? These two example. Motor protein interact with cytoskeleton fragment to promote the movement. This is the good example. Right? Here's another one. Right? You can just mention about these two examples. The invagination of inner mitochondrial membrane are related to explain why. Okay, inner membrane are related to mitochondria. Okay, so can I talk about here? It's because inner membrane is a site ATP will synthesize in mitochondria. So okay, so in the membrane they have a structure, several layers. They increase the surface area of the inner membranes. It increase their capability for ATP synthesis, right? It supplies cells with more usable energy because when those kind of like cellular respiration happened, it's happened in the inner membranes, right? Inner membranes, there's a lot of the protein and the enzymes. They have to convert the glucose into the ATP, right? Because inner membranes, they have several layers which increase their surface area. So this kind of the increased surface areas will help the get more exposed to a protein and the enzymes, and then this reaction gonna and it will help speed up the reactions. Use their knowledge from cell biology of fabric company want to create a new material. A miniature stable and be a high level attention. What several functions might use in plastic? Okay. Okay, go to the attention. It should be E, both B and D, right? Because high level of intention, right? Because this collagen is some kind of protein. It's a collagen is some type of a protein which have strong like tension. Like for example, our hair it's made of a collagen. Collagen is a protein. Our hair can extend, right? It's can extend. It's not like fragile, easy to break collagen. And then this intermediate filament, because filament it can move, right? It can stretch. That's why filament can move because it's stretch. That's why we use B and D. But for elastin, this is a fabric to create material to be a high level. Oh, this A and C, microtubes. Microtubes, it's more like organized, like a organelle. It's like remember that picture? It's microtubes are made by like several proteins they stack together, so it's more like rigid. It's doesn't have like the tension. It's not flexible. That's why it cannot be used for material which extension. Elastin the same thing. It's very similar on microtubes. It cannot extend. Okay, this one is easy. Radius, right? Radius. So this is using. Let me calculate the notation. So surface area, right? Surface area. It's pi square, right? Volume. It's equal to 3, 4 pi 2, 3. So S V equal to, this is R. Oh no. Okay, this is 1 because we already get rid of this. We already get rid of this. This become 1, right? So this become 4, 3, R. Okay, this is the answer. Okay, so I think it's A. You can calculate it. Let's erase it. How to erase it? I want to erase it. Let me see. I don't know how to erase it. Okay. Okay, a mutation arise. Okay, mutation. Mutation, what's mean? Mutation, which means, like for example, we can talk about like the gene. Like my gene is because I look different from you, so my gene is different from your gene. So like for example, I get my gene from my parents. It should be identical, right? It should be identified. But what if like something happened, like my gene changed? Like mutation is called the change on the gene. That's called mutation. Because and called the right side, maybe change amino acid in the active side. So the enzyme doesn't function. What's the effect mutation? That's true. Okay, should be B, right? Because it says the gene, because enzyme, it's from the gene. Gene was changed, so enzyme was changed. So enzyme doesn't work anymore. This is the enzyme. It's called the lysosyl acid hydrolysis. So hydrolysis, which means this enzyme was used to hydrolysis, do some kind of hydrolysis of some molecule. Then it doesn't happen anymore. So hydrolysis, which help to digest some molecule, right? Now it doesn't work anymore. So which means it cannot digest the molecule. It cannot. This hydrolysis cannot happen. So that is the B, right? Let's see. Let's suppose a mutation protein contain short. Okay. Mutate. Okay. Mutant protein. So protein changed. Protein used to have it's kind of sorting signal. At this target. Okay. It's a sorting signal. It's supposed to go to ER. As well as a sorting signal as carbox and target to mitochondria. Okay. So it's this sorting signal help it go to ER, help it go to the mitochondria. It use fluorescence microscope. Fluorescence, okay. Oh, so basically fluorescent microscope. So basically fluorescent dye. So you put some kind of dye on a protein. So this protein was fluorescence labeled. So you can use the fluorescent microscope. You can see this protein, right? Okay, so it's changed. So it cannot go here. So, okay, so if it didn't change, it can go here, it go to mitochondria. Now it's changed, right? So which means it cannot function anymore. It cannot go here and go mitochondria anymore. So it will be in a it contains sorting signal at its amino end. The target is as well as sorting signal. It's oh, okay, because it's mutate, it's changed. So how to do it? So if it's not it's not a mutate. It's if the protein function normal ways, first it will go to ER, right? And then this protein first it will go to ER, and then in the ER it will finish the synthesis, right? After it's synthesized, it will move to the mitochondria. That's how it could be, right? Because it cannot observe into the cytosol. Why? Because the protein will synthesized in the ER, right? It won't synthesize it in the cytosol, right? So in the cytosol, we cannot see it because it's made in the ER, right? So we can see it in the ER, but it mutate. So which means something happened. It cannot move into the mitochondria anymore. So we cannot see it into the mitochondria. So if it's not mutate, it's not changed. Eventually, it synthesizes into the ER, and then it moves into the mitochondria. We should be able to see most of the protein in the mitochondria if it didn't change, right? But right now it's changed, so we'll stay in the ER because it changed. It cannot move into mitochondria, so we can only see it in the ER. That's why B is the right one. Got it, right? Observation, not because see the evidence. And okay. Okay. Should be C, right? This series is talking about how the genomes in the mitochondria and the chloroplast is coming from, right? It's coming from like the bacteria, right? Which go inside through the endocytosis, right? So the first one is right. These two has genomes, which coming from the bacteria genome, right? And then this can divide by binary fission, right? I just talked about how mitochondria was divide, right? First, the chromosome will duplicate, and then binary fission, right? But it doesn't have a mitochondria, right? It doesn't have a ribosome, right? Ribosome only exists in the nucleus, right? So C is it's not considered, right? And let's talk about okay, a molecular biologist catalyze the protein. Okay, protein. So protein is a protein. It's some kind of enzyme protein can degrade the protein. Okay. Patient. So basically, biologists analyzed like skin cell. In that skin cell, there's some type of the protein which can degrade the other protein. Okay, compared this protein, patient protein and skin protein from the other. The number of different protein produced by both types of it's about 900. The number of protein produced only by the cancer. The number of different protein produced by both cell type. It's about number of proteins produced only by cancer. What? Produced or number of proteins only by the normal cells. Okay. Mutation can cause some genes. Some genes to be present, and some genes are not. I like some of this difference in gene expression. I like to come to the case of the mutation and change in the genome. Okay, so what's the difference between cancer cell and the regular cell? Cancer cell, because cancer cell is some kind of like cells which function abnormally. So most times cancer cells, why normal cell can convert to a cancer cell? Because that's a mutation in the genes, right? And then that's why they're different, because cancer cells, they can express 37 protein, and then normal cells, they can express 22. The number is different, right? So which means the gene is different. The gene in the cancer cell is different from the regular cells. That's why they're saying, then which means the gene in the cancer cell was mutated. Let's say there's a mutation in the cancer cells, cause some genes to express, and then this 22 is normal cell didn't express, right? Because the different number of protein expressed differently. That's why. Because during the cell growth, a lot of proteins are involved, right? The number of the protein existing in the cancer cells and normal cell are different. So which means cancer cell and the normal cell, they will grow differently, right? Because they have a different type and different number of the cells. That's why cancer cells grow quickly and normal cells, they don't grow that quickly, because cancer cells, they have more number of protein involved. So, and then they will grow more. That's the difference between cancer cell and the regular cells, because they grow, the way they grow are differently. The reason why they are growing differently, because cancer cells, they have more proteins involved, right? More protein involved, grow more quickly. That's the reason. I suppose you are able to inject a large particle. Suppose you are able to inject a large particle still with a large time. Yeah. Okay. The gauge. Right, too large to take into the memory vehicle. Following the injection, you observe. Oh. Oh, I didn't mention about the cis and transcultures. Let's talk about this cis and transculture. I forgot to mention about this. I think Google. It's quick. Right. Oh. Oh, okay. Let's do this. So this is Gauchi. Okay. That's cis. Cis is here. Trans is here, right? What's different between cis phase Gauchi and the trans phase Gauchi? Cis phase Gauchi. So receiving phase of the Gauchi body to reach the vehicle from the ER empty, to reach the vehicle from the empty their comment. Okay, so basically, so if there's some kind of vehicle here, vehicle is from ER, right? Within the vehicle, there's some kind of a protein or molecule inside, right? The first vehicle will come to cis, right? And then it's received immature or unmodified protein, and then the trans, this part, existing phase of Golgi which will leave the Golgi body. Okay, so first, from ER is somewhere. Okay, from some kind of ER, like for example, some protein, they go through ER, they did some kind of modification, and then they leave the ER, and then this kind of modified protein will be put into some kind of vehicle, and then those kind of vehicle which contain the protein will move the vehicle from the ER to the Golgi, and then the vehicle will arrive cis, right? And then the protein will be released from vehicle and then go inside of this Golgi, and then some kind of further modification will happen for this protein, and then it will move from here. The modified protein will move from here to here, trans, and then during this trans, the protein will be released. Right? That's why it's saying from which vehicle leave the Golgi block facing the cytoplasm, right? Modified protein. That's how it happened, right? Now we understand this. Let's go back to look at that question. Hold on. What's the question? Let's see. So a large particle, okay. The particle with dye. So this red dye will go through cis, and the red particle is too large to be taken into the membrane vehicle. It's too large. For the injection, you observe the location for a long period. Describe what you expect. Okay, so because this red particle is too large, it cannot into the membrane vehicle. Okay, so which means it will stay in the cis, right? Because it cannot it cannot go to the membrane vehicle, so it cannot move, right? So it will stay in a cis Golgi. It cannot move. According to cis measure, it will move. But according to these kind of models, so that model, it says any protein or particle which located into the cis Golgi will be transported into the trans Golgi, will be released away from the Golgi. From that model, it will move from cis to the trans. That's how it happened. Okay, we still have 5 minutes. Let's talk about the memory. Okay, then we're gonna talk about, if some next chapter, we're gonna talk about memory structure, synthesis, and transporter. So in this structure, next chapter, we're gonna talk about memory structure, and why we're saying membrane, it's more like fluid. Which means it's mobile, it can move, right? So which means the memory structure, it's flexible. How to synthesize, like what kind of components within the membrane, right? How to synthesize this during the eukaryotic cells. And then we're going to talk about the membrane transportation, right? So basically talking about how molecule was transported within the membrane, like, right? We're going to talk about the protein which help the ions carry out through the membrane. And then we're going to talk about excytosis and endocytosis. Excytosis, so it's just like release the chemicals from the cell into the outside environment. Endocytosis is like the cells getting the chemicals from outside of the environment inside of the cell. So here he's talking about very, very interesting disease, cystic fibrosis, right? Like for people who have cystic fibrosis, they have a lot of mucus in their lung, so they have a very difficult time to breathe. They cannot breathe normally. right? So this kind of disease, it's all because of some kind of genetic mutation. Like for people who are born have this kind of disease, which means something wrong with their gene, right? It's a genetic disease caused by mutation in a gene called CFTR. That's a gene, right? It's encoded a protein named cystic fibrosis conductance transmembrane regulator, right? Because of this, something happened to this gene, and this protein cannot function normally. And then this protein is very important for digest those kind of mucus within the lung, because this protein cannot function normally, and then the lung cannot normally digest the mucus within the lung, so people cannot breathe. That's how it happens. So this protein, its function in the transport of the chloride ion across the membrane of mesenchymal cell, right? It's a protein, right? So this SCTR, it's a good example of a plasma membrane protein, right? It has to function normally, otherwise it will introduce a lot of disease in us. And the most important thing, why this chapter talking about this membrane, because this protein, because this protein, it's just it's on the membrane. It's a membrane protein. It's like a membrane is like 2 layers. It's this protein is just a how to say, it's a protein just here. It's like this kind of protein here. It's just immersed among this kind of membrane. If this like membrane is very important, right? Because the outside, it can the molecule, it's, if you want to go inside of the cell, it have to go through this kind of membrane. If like inside of a molecule, it have to get out, you need to go through those kind of membrane. So which means if this kind of protein, it's something is wrong. So the molecule cannot exchange between outside and the inside, right? So if those kind of interaction and exchange of the molecule nutrient cannot happen, then which will cause a problem for the cell function, which bring a lot of disease. That's why this disease happened. It's called, right? That's why we need to talk about this membranes, because when we talk about membrane, we have to talk about proteins, which in between.